Hi everybody, I'm Isaac, you probably know me. I'm going to talk for a little while about SB7.0, the 7th Great International Synthetic Biology Conference. I was not at SB7.0, I was in Ohio, it was in Singapore, literally the opposite side of the globe. But because it was the opposite side of the globe, it was 12 hours apart, and they live streamed the whole thing and took questions on Twitter. So I was able to just basically stay up all night, 3 nights in a row, and watch a lot of it. So it was really, really amazing. There were too many things to get into, a whole bunch of detail on, and a lot of the stuff that was presented there in terms of research will probably show up in like later newsreels. So I'll just talk mainly about one particular thing that has come up already, which is the creation of this biotechnology commons. And I want to give some context for this. Basically, one of the rate-limiting obstacles in synthetic biology right now is intellectual property. And that's because the intellectual property framework that works best for synthetic biology is one where you can take established parts cheaply and use them yourself, modify them, recombine them, remix them in a way that is sort of very easy and clean and doesn't involve tons of lawyers. Now the problem is that the IP framework for biology now is based on sort of the first wave of biotechnology, where it was very difficult to isolate a biomolecule that does something interesting, like a protein. It was very, very difficult once you figured out that interesting protein to find its gene sequence in the genome. And then it was even more difficult to take that gene sequence out of the native organism and put it into a useful model organism to sort of start making tons of it and selling it as your biotechnology product. And because all three of those things were really hard, it made sense to be able to patent individual biological parts, and even to patent organisms that contain a single useful biological part that's been inserted unnaturally into it. And this is still sort of the basis of the entire biopharmaceutical industry, the basis of Monsanto's agricultural products. So it works, the patent-based framework for biotech works well for that model, and it's makes a lot of money. But if you want to be able to make sort of more complex things out of a bunch of different parts that you remix, it becomes very, very expensive. And so Drew Indy and the BioBricks Foundation have been thinking about this for a long time, and they've been trying to make this sort of commons and drawing on inspiration from the open source software and the free software movements, where basically you publish your source code so anyone can use it, or you, in software, you actually use copyright to enforce that anyone who uses your code has to then freely share the code that they have created that uses your code. And so Drew Indy and the BioBricks Foundation and other people have been trying to do this for a while. The first iteration of that was actually the iGEM registry. They take all of the parts that the college kids make in the iGEM competition and they put them in the iGEM registry and they transfer it through this material transfer agreement that ensures that nobody will get sued for using those parts if they get them from the BioBrick registry. And so this is the evolution of that, but it's sort of a massive step forward because instead of sort of slowly, incrementally adding in whatever parts college students working over a summer produce, this is creating a gigantic sort of 10,000 gene commons, and it's also using a new material transfer agreement that allows distribution not just to academic centers, but also to small community labs, to companies, and enables people to then commercialize products that they make from recombining these parts. So basically, it's really, really cool, and I am super excited for it. And I think one of the coolest parts of it is that we get to choose what goes in it. They're going to create a forum. It's not clear how the forum is going to be set up. It might be a subreddit, actually. They have one reserved already. It's called Bionet, where people will upvote suggestions for what should go in the registry. And the ones that get upvoted the most will get synthesized first. And BioBricks will pay for the whole synthesis and will basically distribute these genes to anyone who wants them free, plus like maybe the cost of shipping. So can I just get a quick head count? how many people would be interested in helping to brainstorm what should go in this registry? All right, decent number of people. Cool. Yeah, we're definitely going to be doing that. So we'll find you. Yeah, that was the coolest thing I saw at SB7.0.
So one of the sessions at SEED this year, which is one of the big synthetic biology conferences, was on T-cell engineering. So the current state of the art in T-cell engineering is something we call chimeric antigen receptor modified T-cells. So in this design, you use an antibody on the outside of the native T-cell receptor to target antigens that are specific, or mostly specific, to tumor cells. When that antibody binds to the antigen on the tumor cell, it engages the receptor, causing the modified T cell to kill this cancer cell. Or at least that's the theory. It works great for blood-borne cancers, and we're having some trouble translating it to solid tumors. It's got some new both medical and engineering challenges for the field. So the first big one is that one antigen might not provide enough specificity. And so you can imagine that anything a tumor cell is going to be expressing, something else in your body might be expressing. And so this leads to your T cells killing something maybe in your liver or your heart, and that's not great. So we call that on target because it's binding the right antigen, but off tissue because it's not in the tumor. And so everybody is kind of working on this problem, but the two big talks this year were from Wilson Wong's lab at BU, and he uses this, what he calls a supercar system. This is pulled from his patent on it because it's not quite published yet, where the receptor itself is capable of Boolean logic. Instead of doing it at the transcriptional level, like most people do when they multiplex receptors, and is difficult, he's done it extracellularly through some cool protein engineering tricks with leucine zippers. So he's made a bunch of them, but in this one example, there's an AND gate. So you have your first antigen here and your second antigen here, and they have antibodies attached to leucine zippers. And the theory is that the leucine zipper interaction with the T cell receptor is so weak that you need both of them to engage the receptor and hold it clustered so that the T cell can activate. The other one is out of Yvonne Chin's lab at UCLA, and she uses this strategy called covert. So tumor cells also upregulate proteases and other genes. And so their theory was, maybe we could use this property instead of looking for more antigens. And so the way this works is the enzyme that kills cells that is delivered by the T cells is something called granzyme B. And so they took a form of granzyme B that's inactive and separated the inactivating domain, the inhibitory domain from the rest of the enzyme. They put it together with a substrate for a protease that is overexpressed in tumor cells. So the theory is if this gets delivered, if a T cell tries to kill a not tumor cell, the granzyme B goes in there and it can't be activated. But if it tries to kill a tumor cell, it goes in there and is activated. And this works a little bit. It was published in ACS SynBio this past year, and they're currently actually working on replacing like the native granzyme B using Cas9 with their engineered one. So the second challenge is the immunosuppressive tumor microenvironment. It was really impeding T cell function. So you can imagine the tumor doesn't want to be killed by T cells, so it produces all sorts of molecules that are like, hey, don't kill me. And it works pretty well. So a lot of people have worked on this, and again, Yvonne's not published this work yet, so all I have is her the description of her patent from the UCL website here. But basically, they've been able to design a CAR against one of these immunosuppressive molecules, so if the tumor tries to signal, hey, stop killing me, the T cell interprets that as, hey, kill harder. This works really well. It's cool stuff. And the last one is this new one called antigen escape. So this is a normal biologic phenomenon. If you apply immune pressure against a specific antigen, cells that express less of that antigen will survive and become the dominant population. It's a natural selection theory for tumors. And one place we really see this is in leukemias and lymphomas, where there's two antigens that these B cells express. They express CD19 or CD20. And the functions of those two molecules are slightly redundant. So if you design a T cell against CD19, which is the state of the art for B cell leukemias, you wipe out your CD19 positive B cell population. The problem is, then these tumors recur, but this time they're CD20 positive. So imagine if you had a T cell that was capable of killing both CD19 and CD20 positive B cells, that this would prevent the immune escape phenomenon. So this here is a normal CAR. It's got one antibody attached to the outside, so it can recognize one antigen. This is a bispecific CAR, so it's got two places where it can bind antigen. So it can bind CD19 here or CD20 here. And in a mouse model of leukemia, where they study this population relapse and immune escape phenomenon, it was able to extend mean survival by about a week using the bispecific CARs as opposed to the monospecific one. And so this is actually probably enough data that she's going to take this into preclinical trials, which is pretty cool. So that's it for me. Thanks. Yes, Isaac.
Are there other tissues in the body that produce like these immune suppressive signals that the chimeric antigen cells might?
Yes, and she has them multiplexed in a way that you need both the initial antigen and the second immunosuppressive signal to get full activation.
Cool. So another great talk at Seed was a keynote from David Baker. Of course, any talk from David Baker is going to be, every slide is something that is or is about to be published in Science. So it's a little humiliating in that way. But essentially the takeaway is that really what synthetic biologists have been doing, which is taking proteins from nature and trying to repurpose them for what we want to do. It's very primitive, and it is where it's sort of Neanderthal-esque. What we really should be doing is what they're doing, which is designing proteins specifically for our purpose. So a few highlights from what they're working on. They have, as Adam already stole my thunder a little bit, these trimerized HA and botulinum toxin binders. So you have these trimers that bind to 3 different antigens of the toxin. You can get really good neutralization of flu and other things. And then another really interesting thing they're talking about is this proteins that are held together by central arrays of hydrogen bonding networks. And so this is sort of similar to, or analogous to what you see with DNA, where you have Swanson click base pairing. But with this, you can now add multiple strands. You can have 1, 3, 4 strands, or as many as you want. And they're doing some cool things with this, like adding hydrophobic patches that these things incorporate into the membrane. You can basically start designing like transporters and stuff like that with this type of strategy. And they also have a lot of cool things like allosteric protein switches that cause these trimerized binders to switch to a binding state just by adding a peptide key. And they're doing some interesting work designing these nucleocapsid proteins and trying to evolve there. Another great talk was about synthetic epigenetics. This was from Ahmad Khalil at BU. The essential idea here is to create memory by using chromatin regulators to basically write and erase DNA M6A methylations. And these things can be engineered with zinc finger binding domains that recruit your modification enzymes to specific DNA sequences. You can add in M6A modifications or remove them, and then engineer readers that essentially translate those modifications into a M6A dependent gene expression activation, stuff like that. So that's just an example there where they have background where you see low gene expression, and then when you add your chromatin modification, you get increased expression from that BP16 you just going to see. That's about it.
Hi guys, I'm Weston. I'll be the last seed speaker, so I want to go over a few things that I saw that were particularly exciting. One was a presentation by Oxitec, so a lot of you are probably familiar with them, but they are basically interested in the genetic control of insect pests, in particular mosquitoes, and they're actually the furthest along in this. There's several companies who are attempting to do this with things like gene drives, but Oxitec is actually getting quite far in that they're actually doing trials at this point. So it was a good update to hear from industry. The stuff may be available, but I haven't been able to find that much on the news. But what really struck me, and so they're out of Oxford, and so they're out of the UK. But what struck me is that it's not just mosquitoes; they're actually targeting like a whole slew of various agricultural and public health pests. And so these are like medfly, olive fly, like all kinds of moths, all different kinds of mosquitoes, and like sable flies. So it's actually they're trying to build this entire like company around basically suppressing these populations of pests. And what's interesting about their technology is that it's inherently self-limiting, and they made a big to-do about that, so which is accurate. So I think that this might be like the synthetic biology solution we need right now. And so the idea is that they engineer male mosquitoes and they grow them actually in the presence of, I think it's actually TED, right? It's actually like a TED repressor, and they grow them in the presence of that. And so they're fine when they grow, but then they release them into the wild, and then they can mate with females, and then their offspring don't have that TED environment, and then they basically die during development. So and females only mate once, so this is a way to like suppress the population. But if you stop releasing mosquitoes, then your population will bounce back, hypothetically. I mean, population crashes are a thing, so I would argue that it's not entirely reversible, but certainly not anything like gene drive. So it's important to know that like these kind of technologies are out there. And they threw out some pretty impressive statistics, like they did a Cayman Islands trial in 2009, which I didn't even know things were happening that early. 96% suppression of the target mosquito population. A lot of this is with the yellow fever mosquito. They did a trial in Panama. They've done a number of trials in Brazil, over 90% in all cases. And they have production facilities where they make like 60 million of these male mosquitoes a week, which is like just a pretty amazing technological marvel. It sounds like counterproductive, right? But and then I guess really critical for the male mosquitoes thing, or for the mosquitoes thing, is that you can sort males and females by size. But but that's not true of all insects. So they actually had to come up with this like workaround where instead they target the females, and then the female offspring die also later. That one, so they had an interesting genetic workaround for that. But then they also have like a bunch of public education efforts. There's like friendly 80s or something like that. I don't actually know how you pronounce that name, but they have like they showed like pictures like in the middle of Brazil, like these booths with like this like mascot who runs around and like tries to be a friendly mosquito. It seems like mixed signals to me. But yeah, so they are definitely working on that. Monroe County, which I think is in Florida, I think it's actually the Florida Keys, has approved basically 31 out of 33 precincts or whatever have approved the use of Oxitec technology to fight Zika virus. So this is coming to the U.S. Soon, so that's cool. So we talked a little bit about Twist bioscience, data storage a little bit, but what's fun about conferences is you can actually like corner people and like ask them questions. So Emily LaProuse was there, and I actually walked up to her and was like, hey, we're from GMods, and I mentioned how a few members are really interested in using their BioNet system, and then what I really wanted to ask her was, why do DNA data storage? It still hasn't really made sense to me. Like, I'm as much a synthetic biologist as the next guy, but seriously, I could understand if you want to shoot the Library of Congress to the moon, might be helpful, but that seems like an niche application. So, she actually threw out some really impressive ideas, and so this is really helpful for me when I think about this. So apparently oil fields, when they do seismic surveys of them, those surveys are worth like hundreds of millions of dollars, okay? And then they keep those oil fields for like hundreds of years. So they need that technology, or that information as a backup to last like hundreds of years. And so if they would lose that when hard drives get corrupted, if that gets corrupted, then like you have to resurvey your entire oil field. So they keep mining the same field. So apparently that's like itself a multi-million-dollar industry. Medical record backups are actually legally required, and if you like would lose those, you're actually breaking the law, so there's good reason to have several backups for those. Apparently, she talked to James Cameron, who's making Avatar 2, which is coming out, and he wanted the audio files, like the really rich audio files that it takes to make movies, right? Actually, gigs and gigs, right? And he's like, okay, team, get out the Avatar 1 audio files, and they're like, we don't have them because their hard drive crashed. So he was really pissed off about that, and now wants to use DNA data storage, I guess. It's kind of fun. And then there's a big market in defense-related data storage. So DNA, because it's physical and like not connected to the internet in any way, can't be hacked. So that's kind of cool. And then it isn't destroyed by like electromagnetic pulses. So you can imagine like there's like a hydrogen bomb, like half the United States has all the hard drives are gone, right? So your DNA would not be affected by this. It's like in a lead case. And so you still have to get it to a sequencer, but assuming you can get it there, get this cataclysm. But anyway, it's an interesting idea. They'll probably sell some stuff that way. Microsoft is certainly investing like millions and millions of dollars. So I hope Microsoft isn't stupid, but it still remains to be seen. But anyway, just like to put some meat behind that. That was a lot of fun talking about that. And they have these cool acronyms. So apparently there's like a whole class of data called worse and warn that like basically never gets read, but you still have to have it because reasons. The other thing I wanted to highlight was a few talks I thought were just like really good. They're like not necessarily what you would call like the keynotes, but I thought they were really great. One is on, so there's a whole section on engineering the microbiome, which I think we're getting further on than I thought we were. So there's a group from University of Minnesota who's working on microbial delivery of antimicrobial peptides. So you engineer a bug, then you to produce antimicrobial peptides, then you ingest that, and then it like shoots them around to the drug-resistant bacteria in your gut and can therefore treat. And that's cool because antimicrobial peptides can be much more surgical than standard antibiotics. So, and also a lot of them don't have resistances yet, and there's a lot of them, so you could use that. There's also a really big market in the poultry industry because you could be antibiotic-free but use antimicrobial peptides. It's a cool trick. And you're not modifying the chicken, but you're putting in a modified bug. Wow. So that'll probably hit Whole Foods at some point. Or Amazon. SynLogic also had a cool talk. They have a whole variety of metabolic disorders that they're trying to engineer bugs to treat. Usually you ingest these bugs and then they inhabit your gut somewhat. The issue is colonization, and that has been the issue for a long time, but they're working on that. And it can basically do what your body can. So whether that's producing butyrate or like digesting arginine or like a variety of other things, it's kind of a cool idea. And then they have this whole thing, it reminds me, does anyone remember like this paper where they were like, we can treat cancer by injecting like E. coli that go kill the cancer, right? And you read it and you're like, that's called sepsis. But they're trying it, so they're really excited about it. And I guess they say that their E. coli like only live where the tumors are, so they're friendly and all this stuff. It's still gonna be a while before other people like shoot live bacteria into my bloodstream. But anyway, definitely they have a large arm in that area. And then what I actually thought was the best talk, and unfortunately it's still unpublished, but just an incredible talk by Michelle Chang at UCal at Berkeley. And it's basically this idea with metabolic engineering to bioorthogonal chemistry. So there actually are some bugs that produce alkyne amino acids. So if you're familiar with like click chemistry, so these are alkynes and azides, you can do click chemistry with those. And what would be really fun is if you could make your bugs produce your bioorthogonal chemistry. So have like naturally occurring non-standard amino acids, right? So in this way, you could produce actually your non-standard amino acid, which could be useful for bioorthogonal chemistry, but have it be made in the bug rather than like adding it in the media. So anyway, she did an incredible, basically like really chemistry heavy, really mass spec heavy exploration of this pathway, and I do think that they'll be able to put it into E. coli soon. So, so it was a lot of fun. That's all I have. Any more questions for me or the other speakers? Yeah. Yeah, so the workhorse here is Nissl. So E. coli, Nissl, 1917, isolated World War I-ish time period. So yeah, that's what they're using. And it seems like they're kind of settling on that chassis at this point. But they're using it to treat, salmonella is one of the things that they like treat and various other bugs. But yeah. Cool. All right, hell yeah.
So for Ocutex, if people who haven't seen it, on their website they have these amazing like promo videos of like technology they're food. So
Cool. Yeah, it's really fun.
Play animations, mosquitos.
Really? Awesome. Yeah, I just want one of those like costumes. right? Like their mascot. It'd be a great Halloween costume. Be an architect mosquito. All right. Well, thanks guys. Stay tuned for more updates. All right. Stay tuned for more updates from GMODs. Definitely listen to the podcast. It's incredible. And then we'll have a meeting next month. So thanks. No, I'm just really worried.
I feel like that also, like, if you're doing that,
Backfire.